we will get started. And I'll do my YouTube introduction. Listen, when we get up to 30 viewers, I want to just claim that I'm YouTube famous at that point. No, that's never gonna, unless I do something really embarrassing in front of everybody. That won't. Gotta go viral. Welcome back to day 2, part 2, where we are going to be talking about binning. And so I didn't mention this on the last slide, and I had mentioned it yesterday, but I should be mentioning it each time because who knows where you're coming into this if you're watching this on the internet. A lot of this information has been this collaborative effort that's been happening over multiple iterations. Using my original slides, people who ran the EcoGeo aspect of the workshop in 2016 added their own flavor to it. I've now reabsorbed their flavor and I've been working with that too. So the last set of slides that we worked with for assembly had been co-opted and made better by Frank Alwood and Dan Mende at the University of Hawaii. This binning step that we're going to talk about comes from the brain of Murat Eren at the University of Chicago. And we're going to be using his tool, Anvi'o, to visualize the binning aspect as we go forward. So this will be a little bit of a very soft Anvi'o tutorial, but also like Anvi'o is great, but what if we could use other data going into Anvi'o if that's something that you're interested in. So I'll start with the presentation here, and then we'll get to the part where we get to go hands-on, and we'll talk about binning. So why are we binning, right? So this is this goes back to why are we doing metagenomics in the first place? And we are doing this so that we can access the genomes of organisms that haven't yet been cultivated. So it's a very positive outlook on this, that someday we'll get there and we'll cultivate them. And so there are a couple steps that go into this. And so we need to get our genomes from somewhere. So we have to take our samples. We need to generate our sequences, quality control them. We need to go through some assembly step, which we just covered. And then we need to go into this binning step to try and recover genomes. And so you can see here in this little schematic where we have colors representing the starting genomes and the output genomes. Even in the most beneficial, positive environments, a lot of times when we do a binning and get an output from it, we don't get the full genome, right? That's represented here by these fragments. If we get really lucky, we can, but it's not always a guarantee, and that's okay. So this would be an environment where okay, so these are where all the organisms are present at equal abundance, and we can see them represented equally in the short read output generated from the genomes. But in reality, environments are much more messy than that. And so we can actually end up in a situation that's a little bit more like this, where we have some starting community, and then for whatever reason, either because it actually is more abundant, as in the case here, or because of some artifact due to the library preparation process or the sequencing process, some organism becomes more dominant. And so they become dominant in our short read data set that we're working with. They become dominant in our output of the assembly. And then because they're the dominant organism, we get the most of their genome back and maybe only fragments of the other organisms back. But that's okay. We're going to still work with that. And the goal is to take these fragments that we have that are colored in this way with the contigs and group them together and make it so that they all belong together, right? So we can get all the yellows with the yellows and all the greens with the greens and have some estimate of a partially complete, near complete genome of an organism from the environment. The problem gets much harder if you take away the identity before you get to that step, right? So while the colors, when you can see the colors, are really nice and you can be like, yeah, yellow goes with yellow, it's easy. As soon as you take away the colors, you're like, okay, this is a joke, right? How are we supposed to actually do this? There's no information here. How are we gonna do this? It'd be like looking at a puzzle that was just all black, right? And then the sides changed as you were working with them too. So that would be really problematic. And so there's a lot of information in DNA, and it's been something that we've known about for a really long time. So this is a paper from the 1960s where we talk about how if you look at the products of DNA synthesis, there are unique patterns depending on the organisms that you're looking at based off of frequency of the occurrence of certain base pairs. And so again, this is something that we've, so that was in the 60s, in the late 90s, we were able to do this too. So this is a comparison of 9 genomes, and we're looking at their dinucleotide composition. And so tetranucleotides is something that we work with fairly commonly, which are words of 4, but in this case, we're looking at words of 2. So we're taking the genome and we're breaking them up into words of 2 letters, right? So the A's and T's, C's and G's, and we're comparing them and seeing the occurrence, right? So if we map every one of those possibilities and put it into like 2D space, can we map and see where these organisms map out? And so we get something, so this is one of the figures from the paper, we get something that looks like this. And so each of the lines on these circles here represents a different base pairing. And we can see that if we ask how often do certain base pairs occur for different organisms, so in this case, E. coli, comparing it to influenza and Helicobacter pylori, you can see that these star shapes actually look different, right? So E. coli has one star shape, Helicobacter has a different one, the Seneca cystis has another shape, right? And if we were to map that using, going forward and mapping it with tetranucleotides, I'm getting ahead of myself here on this one. Next slide after this. We can start seeing these patterns of tetranucleotide frequency along a genome. And so that's what we have here. Each of these, so along this space right here, we have the genome, and then along the y-axis, we have different tetranucleotides, so 4-letter combinations, right? So of the genome, and in this case, it's a heat map where red indicates that there's more occurrences of that particular tetranucleotide versus another one, and then highlighted here in a couple of spots. So these stars and this large black area actually represent tRNAs and rRNAs. And so we actually can see a shift in the tetranucleotide frequency at certain points within a genome that we're looking at. And so if we map this, so this is what I was getting to, if we map this onto 2D space and look at tetranucleotide frequencies and compare things, you might be able to do this a little bit by eye, depending on which organisms you're working with, right? So this Archaeoglobus up here has one pattern of tetranucleotide frequency based off of, so here's GC content, here's some, or here's GC content, here's some variance in the tetranucleotide frequency. So if you were to overlap Archaeoglobus and the Helicobacter here, Archaeoglobus occupies a real different area of this map or this plot than the Helicobacter does. So if you had a bunch of fragments from either of these two organisms, and you looked and mapped their GC content to their tetranucleotide variants, you could actually maybe pull these apart. You could even probably do it by hand. You could look at it. There might be some overlap between this region and this region, but you could actually tease apart the majority of the Archaeoglobus and the majority of the Helicobacter organism. Compare that to E. coli versus Archaeoglobus, or Archaeoglobus and Seneca cystis. They basically lie on top of each other, right? And so while there is information that we can tease apart when it comes to tetranucleotide information, it's not 100% discerning between different organisms. And so if we got into a situation where we had fragments of Archaeoglobus comparing to fragments of Seneca cystis, and we were saying, hey, can you tease these apart? Using this methodology, you wouldn't be able to do so.
There was a time when that was all the genomes we had.
Yeah.
Let's point that out.
And that's why they did a study and got a really great paper out of it. Okay, so we're starting with contigs that we don't know the identity of, and we use genomic signatures to predict where they might belong, right, in terms of grouping together. And so if we're using something like tetranucleotide frequency, GC content, I talked yesterday about amino acid usage, you might be able to do something like this, where you can pull out some grouping of contigs that works but isn't perfect, right? So we have some overlap that happens that can be pretty, some problematic areas, right? So we have mostly yellow with a purple sticking in here. We have mostly purple with a yellow and a purple. I guess this is magenta, not purple. Purple and green. Green is missing some of the fragments that it should have, right? So we can do okay, and we can get pretty close using these genomic signature methods. And one of the solutions that has really kind of popped up from a bunch of different labs almost simultaneously was this idea that, and I mentioned this yesterday, is that organisms, a genome of an organism is connected to each other. And if you look at for an organism in multiple different samples and use the information that you get from your sequencing run back with the reads and ask, okay, can we find contigs that we've assembled that have similar coverage patterns over multiple samples, we actually might be able to figure out which contigs belong to each other. And so coverage, like I said in the last section, is this idea. And so in this case, it's a specific example of figuring out how often a single base pair along a contig is covered by the reads, right? So that's what this X coverage means here. So it's a little different than the reads per base pair idea. And so if you were to draw a straight line, so if you imagine there's a base pair here and draw a straight line, there's 1, 2, 3, 4, 5, 6. So this is approximately 7. There's like a 7th one here. So if I had drawn it a little bit over, right? So it's 7X coverage for this contig from this sample. And then this contig, it's 4X coverage. So if you draw a straight line, you hit 1, 2, 3, 4, right? So that's what we mean by coverage here. And so if we have a setup like this, where we have 3 organisms, and in each sample, the organisms have slightly different coverage values or abundance values, right? So in this case, purple is dominant, and then not dominant in samples 2 and 3, and green is dominant in 2, but not 1 and 3. And we ask what are their coverage values in these different samples, we can make a matrix like this and ask which ones actually belong together, right? So we have A and E coming from purple. In sample 1, A is 8X coverage, E is 8X coverage, and in the next couple samples, they're 1X coverage. And so we can make these plots and say, okay, look, A and E have the same occurrence values in multiple samples. B and D, I have to cheat on that one. B and D have the same values, and then C and F do. These probably are contigs that are coming from organisms that have, at some point, these are connected fragments of DNA because they all have very similar coverage values in multiple different samples. And unlike the last time where we made it gray and you couldn't really tell the difference where things are supposed to go, it doesn't matter that we don't know where it is started from to begin with, right? The lack of color shares doesn't change, the math still works, and you're able to plot contigs that belong together. So this is what it looked like with genomic signatures. What we can do is we can add in differential coverage from different samples to correct those small errors that we're seeing on the output and get something closer to a little bit more pure and a little more clean draft genome of an organism. And this is happening a lot now, right? So we covered this a little bit in the experimental design yesterday. So starting from metagenomics in 2004 and coming through to the first extracted genome from metagenome in 2012, we're now at this point where thousands of genomes can be pulled out of fairly large data sets using these methodologies. And so there's a couple of different ways of doing this. These are automated programs that will work to bin your genomic data using these methodologies, using both tetranucleotide frequencies, GC content, coverage information. Some of them do different things. Look at these parameters at different points in times in terms of when the algorithm is running, and then also uses different underlying algorithms to make groupings. So one example would be Metabat. Metabat right now is seeing a pretty good boost. So the original paper, which I have here, which came out in 2015, had 1 degree of success. They've actually released a new version of it, Metabat version 2.0, which doesn't have a paper yet, but has, they've corrected some of their issues that they were working with, and now is actually one of the best, if not the best, binning methods. And that's not to undersell the other binning method that's on the slide here, which has been Sanity, which is our binning method. Elena was the one who wrote the code for this and did the paper, and it's a published paper in PeerJ, which also does the same thing, which takes your input data using coverage values and your tetranucleotide frequencies, and in a automated manner will generate genomic bins for you. And so once you have a bin, that's cool, that's great, it's lots of fun. But right now there's not too many ways of working with that data in a visual manner. And so one of the platforms that is out there that actually does that is a platform called Anvi'o. And that's why Marin was the one who did this presentation at EcoGeo a couple years ago. It's because that's his software. And so Anvi'o is a visualization omics platform that has a lot of functions built into it, and allows you to do lots of actually really cool and interesting aspects to it. And so one of the things that we're going to look at is we're going to look at some bins that we've created using a data set from an infant gut microbiome data set. And we're going to look at it through the lens of a couple different binning methods in Anvi'o in a visual manner, so that way we can get a better idea of what this actually looks like. And think about what the data means. If you go to these links that are here, Anvi'o is a really strong tool. It's in its own right, it is a binning methodology in itself. It actually incorporates another third binning algorithm called Concoct. And then they have all of these recommendations about how to use Concoct and Anvi'o together to manually go in and refine the data that you're getting out the bins to make the best bins that you can. And so there's all sorts of elements within Anvi'o that allows you to do that. They have a pangenomic workflow, which we won't have time to talk about, which works really well. But it gives you a lot of options. And so, like I said, we're going to be using, so this has been a data set that has been around now for about 5 years. So this is an infant gut metagenome where we have this fairly simple system as of looking at premature infants, I think they're premature, and watching their microbial gut community develop over time. And so it starts off fairly simply and becomes more complex as they persist in the world for longer. I don't do humans. I don't know what they do at that point. So you have this element of a natural progression of these of this environment, of the samples, where you start off with one handful of organisms that then change over time. And it's an ideal way of looking at how relative abundance as it changes gives you a better idea of doing the binning. So it's nice. The original paper did all of their binning by hand. That's how simple it was. And it allows, because they did it by hand and took a lot of time to do it, it allows some comparison of the automated methods. And so as part of the Binsanity paper, we took that data set, compared it with our assembler or our binning software and a bunch of other binning softwares. And so that's the data that we're going to look at here. Okay, so I'm going to get out of the presentation mode and we're going to start playing around with the data. So the first thing that we want to highlight here is that the actual raw data is not present in the virtual machine. So these are normally stored as multiple. What is going on? Come on, go down. There we go. Start here. So if we're in the EcoGeo folder, we're going to go into the binning subfolder. And if we hit LS, there's a lot of information here. And so like I was saying, there's missing data. So in that 00 BAM files and contigs folder, there's currently nothing stored. That would be where your original raw data is. And so in this situation, you're generating BAM files. So you're, because you're using differential binning here with different coverages, you're going to have been recruiting your reads against your assembled contigs to generate these coverage profiles. So that's not here right now. They're going to be not there. It took up, nobody wanted to download an extra 60 gigabytes of data on their virtual machine, right? Like that's the kind of level that we were working with when we said, no, we're not going to put that in here. Let me make this just a little bigger. Okay, but we can move on. So this has been pre-computed for us. And so we can look at the profiles that have been generated. So if we move into CD into 01, which is the Anvi'o merge profile, and hit LS, there's a lot of information in here. So this is the outputs that are generated from running the initial steps of Anvi'o. When you take, so all you're doing is taking your contigs and you're mapping them using abundance, and it generates using hierarchical clustering, it will generate a tree that you can look at and visualize the information based off of coverage information. So that's what we're going to do first. We're just going to visualize just what the raw information looks like. All right, so we can do that by using the following command. So this is anv -interactive -p profile.db -c contigs.db. Make this bigger too. And if you hit enter at that point, which you can all do, you're gonna see this like stuff happen. And then it should take over. If you haven't opened up your Chrome, it will open up Chrome. If you have Chrome open, it'll take over it. And it's gonna draw the data. So I'm gonna go back and forth between making this big and making it small so we can work with this together. All right, and so this is what Anvi'o, the map of Anvi'o looks like. And so what we've done, what happened within Anvi'o in the profiling step is it took all the context, it calculated all this information in terms of tetranucleotide frequency, and then more importantly, coverage, right? So each one of these layers in this donut circle right here is a different day that the infant gut microbiome was sampled. And then create, like I said, a tree based off of hierarchical clustering. And so you can see there are these, I can zoom in. I'm going to zoom in Anvi'o. So if I zoom in, you can see that there are these groups based on the tree. So I can highlight it here that have coverage in all of the samples. Right, so I've clicked on this and highlighted it. And so you can see that these contigs represented by this part of the tree all have some amount of coverage in all of the samples. And if I were to move over just a little bit here, I'm going to highlight this in a different color. Here. No, we want this whole thing. Highlight this whole area here. You can see that this area highlighted right now actually has coverage in only a few samples, and these samples are the ones up here in the top, right? And so very empirically, if we were just looking at this, we could start to say something about, based off of these coverage values, that these are probably some type of organism that's, the contigs are co-occurring with each other, potentially a genome that belongs, linked in pieces, right? And you'd get into finer details here. So the colors are all going to be one for right now because it's all set. I'm not actually setting different bins, but we'll do that in a second. But I can unhighlight this. So this right here, this is really a clean example, right? These contigs are present in all of the samples at really high abundance, right? And so you can, I'm pretty sure, I'm almost 100% positive that when we go forward, this is going to be predicted to be a single genome, right? And we've just done that visually looking at this, right? So once you get to this data in a visual format, it becomes much easier sometimes to visualize things or to pull out the pieces really quickly. Obviously, you can think in your head of an example of where this might become really cumbersome, right? So in this simple system, we're only talking about 20 genomes. If we start to get into hundreds of genomes, this unless you have a lot of data to work with, it become pretty inhibitive to do this by hand. Not that people don't do it. Okay, so that's our first look at Anvi'o. We can, yep.
What are we visualizing here? Is this one metagenome per day, or is this one bin per?
One metagenome per day.
Okay. So that one metagenome can potentially have multiple, different bins for different facts or whatever. Not sure.
So each metagenome just has the record of what's present and what's not, right? And so as the community shifts, what we're looking at here are changes in the presence and occurrence of certain organisms or certain. Right now, we're just looking at the contig level, and we're going to get into the binning part, right? So the idea here is that whatever these contigs represent on this part of the tree right here, right? They don't have abundance in these samples, these first 8 samples or whatever it is. But in the last 4, all of a sudden they're there, right? And so if you're going on this theory of differential coverage, the idea is that those contigs are pairing together because they're coming from the same source. Okay, and so that's great and how fun, and there's lots of things to do with Anvi'o. And I encourage you to do the tutorial from the Anvi'o side of this. But right now what we're gonna do is we're gonna X out of our current view right here. And what's going to happen, so your screen's going to look like this if you go back to your command line. You're going to try and type stuff, and it's going to yell at you, and there's nothing you can do about it. So the way you get out of this is if you hit hold control, and hit C, it terminates the run of Anvi'o right there. So is everybody able to do that? Yep. Yeah. For Anvi'o. So like if you were, the question is if you were doing this with data that, working on a server and you'd run Anvi'o on the server. Yeah, Anvi'o has a whole setup to show you how to do this, but you can actually directly connect your Chrome ports to the port on the server, and it will send information back and forth, and you can visualize it locally, even though it's on. So unlike Chime 2 yesterday, which doesn't work, the visualizations don't work on the server, there's no direct communication. It's different for Anvi'o. Okay, so that's great. One of the things that we can do before we, again, get into the binning is we can actually, just based off of the contigs themselves, we can predict some taxonomy to assign them, so we can maybe visualize some information about that, which is what we can, and so Anvi'o has this built in. It uses a tool called Centrifuge to look at a database of genomes, which has lots of information in it, and it will compare your contigs to those genomes and assign a taxonomy to it. And so we've done that. We made that calculation already. But what we can do now is we can import that information into Anvi'o and visualize it, and then we can get a better idea. And again, this will be one of those things where, like, sort of at first we could see visually using coverage, that kind of looks like a genome. They all occur together. This will be a very similar thing of, like, not only does it look like a genome, but all those contigs have the same taxonomy. That's going to be really easy to work with, right? And so we're going to do this using the Anvi import command, import taxonomy. So this is one of those ones where if you want, you can copy and paste. This should work. So it's going to be --c big contigs-.db. You're going to do -i, and then we're going to tell it, right? We're going to use commands that we learned yesterday. 2 dots and a slash means look in the folder above us. And then we're going to go into the folder that's called 02 centrifuge files. We're going to use the flag for finding a tab separated file. And then we're going to tell Anvi'o that we use centrifuge to calculate this. And so when we hit enter here, as long as there's no typos, it's going to run. It's going to say how many contigs it found. And because we ran centrifuge separately, it's going to say, well, how many did it had taxonomy? I'm going to assign it to this. Gives you all this information that's happening. And so it says here that the input data from centrifuge annotated 3,000 of the 4,000 contigs that were originally added to this profile. And so now, again, we can go back and visualize this. And so we're going to do this a bunch of times. And so if you want, you can actually hit up twice, right? So at this point, the centrifuge file has modified our profiles database and allows us to, just call the same Anvi interactive command to visualize this. It's going to have like a fun party as it tries to load and draw everything. Okay, and so right now, you'll notice as this happens that you get this little thin line of color at the bottom. That's not going to be very useful for us. So on the side here is all of the information that's displayed on this figure is told what's here and how it's displayed. And so the thing that we're looking at here is this height. So all of the days with the metagenomic data has a height of 180 pixels. And if you get down to the very bottom, we have one called taxonomy, which is a 30. And so that's the reason why it's so small. And so if we change this to something like 200 and then hit draw, which will redraw the figure. Now it's much clearer and you can see it as the colors in the bottom here. So we can zoom in. We can zoom in. And now we've assigned taxonomy to these contigs. And like we hypothesized before when we looked at the data, that these contigs that all had very similar abundance patterns, they actually also have all the same taxonomy. right? Predicted taxonomy. And so you can see the light blue, the magenta, the green, the pink here. And then we get into this area where, for whatever reason, either the database is not sufficient enough, these are novel organisms, or it's something that we haven't seen before. We get into this area where there's some green and some pinks, and then here there's some pinks and a lot of gaps. Here there's a whole bunch of gaps, right, where there's no taxonomy assigned. We can scroll around. We can see this area here, which is poorly defined taxonomically. We have this area here, which has some issues as well. So while taxonomy works really well in this simple system, if you don't have an organism in your reference database to refer to, you're not going to be able to predict any taxonomy. And so while it's a nice guide, it doesn't give you a definitive answer of where a bin is and where a bin isn't. So that's great. We've added taxonomy. It's something that we can look at. It's permanently added to our database. And now we can go back. And so again, we're going to X out of this. We're going to add some more information. We're going to keep working on this. We're going to hit Control C to get out of the Anvi'o environment. We're going to go to the next slide. Okay. So the next slide, we're going to use the folder that has the 03 in front of it. So these are going to be our external binning results. So what we're going to do is, now that we have this data that Anvi'o is generating for us, this nice little visual, we can actually map back the results that we get from all of these other different bidding methods and say, well, how well, what are they predicting? Where do they can look like, and how do they compare to the results that Anvi'o is generating? So this is a handful of tools. So it's going to look at concoct, group them, metabat, maxbin, but version 1, not version 2. MyCC and Binsanity, like I said before, and I'm going to plug again, which is our tool from our lab. Feel free to use it at any point in time. So I'm going to keep adjusting this so we can keep seeing what we're looking at. So what we're going to do is we're going to add the Binsanity results of this of our binning method to the Contig's database in the very same way that similar way that we did to the taxonomy. So we're going to use a different Anvi import tool called Anvi import collection. Dash. So that was dash lowercase c. This is dash big uppercase C. We're going to type Binsanity in all capitals. We're going to do double dash contigs dash mode. So that's just telling you how we're assigning. Anvi'o does this thing where if you have really long contigs, it'll split it. And that's why they're called splits in the last example that I gave. So what we're telling it to do, instead of assigning it per split, we want it to assign per contig. So if there is a long contig that it has split, they'll get the same assignment. That's what contigs mode does. And then again, we're going to look in one directory up for this 03 directory. And we're going to add in the binsanity.txt results. So if we hit enter, it quickly adds all that information about which Binsanity predicted to all of the splits that were part of the profile. And again, if we hit up twice, oh, there's a whole, let's hit a delete here. There you go, whole thing that we couldn't see. So we hit up and go back to Anvi interactive -p profile -c contigs and hit enter. It's going to open it up again. Okay, so again, the taxonomy is here because we had to redraw it. It's still small, so we can adjust that again, make it a 200, hit draw. So that'll increase it. And then what we need to do is we need to tell Anvi'o where we're looking for our bin assignments. And so there's a tab up here called bins, which we can click on. And then we can click on load bin collection, which is what we did with that last command. And in here should be one called Binsanity. Seeing some people who look like they're, yeah, okay. No questions here. We'll hit label. All right, and so now we have all of these bins that have been assigned here to the left. If we go back to the layers tab and scroll down, never mind. That's not what I meant because they're right here in the bins. But if we zoom in, we can look at and see where Binsanity has predicted a bin or a genome compared to where the taxonomy is and the coverage. And so again, this first, very clear bin in blue with this set taxonomy gets assigned to one bin. This here, which has one taxonomy here, actually gets assigned into two bins, right? So they have slightly different coverage values. So it's likely two closely related organisms. This area here, which did not have really good taxonomy, gets effectively assigned to a bin. This area here, and so you can see all of the Binsanity bins going around and around. And so we're able to actually predict bins in areas that do or do not have taxonomy prediction. Sometimes they overlap, sometimes they don't, in a fairly high effective manner here. Okay, so we can actually close out of this. And so now what we're going to do is we'll actually map back all of the results onto this, and we can compare how all of the bins compare to each other. So we can close out, hit Command C to close down. Okay, so now we want to include all of the external binning results into our visualization. And so in here, and this isn't a built-in script, this is actually built into current versions of Anvi'o, but is not in the version that we're using. We actually have to use a script that's in the folder, the directory that we're working with, this Anvi script merge collections. And so we do that by doing / Anvi script merge collections.
There's no module named Anvi'o.
There's no what?
There's no module named Anvi'o.
Module for where?
When you do that.
Just go for it.
Okay, great.
Yeah, you'll see.
Oh boy.
Import error.
Cool. Maybe we'll get to play with our path earlier than I thought. So -C-I. Oh, you know what it is? Because this is a Python script that's independent of Anvi'o. That's what we're going to have to work with. Cool. All right. So this is the solution that we came up for this. So take a little pause right here. Station identification pause. So if we all type echo $path. So everybody's going to need to do this. If you've been doing something else and you're like, I wasn't really paying attention. If you don't do this right now, we won't have the solutions going forward. Okay. So we're going to do echo $path. And so this is what I talked about yesterday. So these are all the folders or directories that your computer looks into when it's trying to, when you do something like run Python, when you're looking for Python modules. And so the culprit here is this first part right here, this miniconda 2. So when we install Chime, we have to install miniconda, and miniconda has decided that it wants to control all of the things, the modules within Python and how they work. So we need to tell the computer to back off. We don't need your help, okay? And so what we can do here is we actually go in and edit our path directly. And so we're going to do that using Nano, our in-screen editor. We're going to put a little tilde here to represent that we want to go back to our base. And then we're going to do tilde / bash rc. And so this is a secret file that contains information about what's in our path. And so this is we're going to get real like matrix text awesomeness here. We're going to hack the system or something. So if we hit enter, it should bring up this. I'm going to make this a little bigger. It's going to bring up all this information about what's happening in our bash. And if we go all the way down to the bottom, so just keep scrolling down until you get to the very, very end, you can see that the miniconda 2 installer has added this information to our path. How fortunate for us. And so because we're done with the Chime 2 elements of this, we don't need to go back to that. What we can do is we can put a # in front of this, and it's going to comment this out and make it so that the computer no longer looks for it. So once you've added the # key there, you can hit Control X. It's going to ask, do you want to save this? You're going to say Y for yes, You're going to hit enter, and that's going to have changed that for us. And so now if we do echo path, we need to type the word source first. It's a source.
Source and then like source the actual file, the bash RC file source, and then write it.
Right.
Yeah.
So we use the command source. Again, we're going to call the bash RC file like that. We're hit enter. And now when we do echo path, it's still there.
Close out of the terminal window.
Okay, let's try that. We're going to close out of the terminal window and open it back up. The easiest way to reset something. Unplug it and plug it back in. So I'm going to make my screen big again. Let's just check it right here. And it's gone. Woo, yay. We can all cheer. It's fine. You can cheer. Thank you, Elena. Oh, how dare you. All right, so if you've closed out of your virtual machine and now we're back here, we need to CD into EcoGeo, into binning, and then we were working in 01, sorry, CD 01. We're working in the Anvi'o merge profile area. So now if we type out Anvi script merge collections -C contigs DB -I star.text collections and hit enter, it works. Everybody, we can thank you, Elena, for saving us all from ourselves. Yeah, that's fine. I'll bring it back up. So it's a -C contigs.db -I /03, and then you can tab out the external binning results. Then we're going to put in the wildcard.txt, and then we're going to output it in something called collections.tsv. Did that work for everybody now? Everybody followed those instructions to the T and it worked. Elena's going to come over and help anybody who has not been able to do this.
You might want to try that.
Why? Why are you doing this to me? Okay. Okay, so let's try running Anvi Interactive that Elena just said there might be a problem with. Okay, not a problem there. -P. Okay, great. -C contigs.db. And now we have to actually call in this collections.tsv file that we just output. So this is going to take all the information from all of the binning output methods that we did before. It's going to add it to our profile. Keep drawing. All right. And so you can see down here in the bottom, if I zoom in, it's like the opposite. Every time it's the opposite of what I wanted to do. I'm going to scroll down. So you can see we've added our taxonomy. And now below that, we have actually all of the bins generated from concoct group of max bin metabat, the original hand created bins, bin sanity, and then mycc. And so if we scroll down here, so right now they're all real tiny. We can change all of these 30s to be bigger, like by making them all 200 or 300, because then you only have to add a 0. So we'll redraw that, and that will increase the size. And then we can compare directly how these different binning methods worked for this data set. We can zoom back in. And right now, all the colors are a little crazy. Our next step will actually correct some of that so that we can maybe visualize a little better. So this first area that I keep coming back to, which has a consistent taxonomy, all of the binning methods were like, yes, this is a bin, this is a genome, this is what you should work with, how great. It starts to fall apart a little bit for this. This one has a little bit more complication, depending on if they're calling it one bin or two bins. Here we have a successful bin, here we have a successful bin. And then we get into this area where it gets a little bit harder, and the binning methods have success and some success and some failures, right? So this binning method here, which I forget the order of, is mostly white here, indicating that it's not accurately binning this particular subset of contigs. This one here has a bleed over, so some of these contigs got assigned to this bin next to it, even though in other binning methods they're predicted to be one organism. We can scroll along. We can see that this binning method didn't correctly assign anything in this area, while this one it has a number of contigs that weren't assigned to the bin. This method here split it into two. I believe that's insanity. And we can go all the way around the circle and look at the results here, where they overlap and where they don't overlap. Okay, so we're going to close out on this one. Going to hit Command C. So we're going to do another import here, again using one of these commands that were generated just for this version of Anvi'o, Anvi import state, and then we're going to import infant gut.state -n. We're going to write colored -p profile. We'll hit enter. And so what this should do is this assigned colors to all the bins. So that way, if this was manually done by Elena, so that way if the bins all lined up, they could all be a set color. So we hit up a couple times, we can go to Anvi interactive -p profile -c contigs -a collections. And we can hit enter, and that will draw a new So again, it's all small down here. We'll zoom in. So the colors have changed. Not exactly how I predicted them to, but the colors have changed to give you a better idea of where the assignments are correct and where the assignments aren't correct. And so this is a file that Elena created by hand that gives you some idea of, right? So you can see the colors a little bit more consistent between and more clearly defined than they were in the last one. Cool. Anybody have any questions right now? I know we're going through this, seeing cool things, we're playing around. Yeah, what did the last command do with the colored?
What is it? Oh, it just so the JSON file is basically like when we were originally doing this, there was no automatic way to do it, so we actually had to manually go in and make sure all the bins that were overlapping had the same colors. So like we manually like basically set it so that each color is the same for corresponding bins.
Right.
After doing a comparison, so that's just like the JSON file is just like the file that holds all the information about what color those basically import results.
Yeah. And so I think now this is done automatically as part of Anvi'o. So after we highlighted it and talked to Marion about it, they actually, in the more recent versions of Anvi'o, that does this automatically. But in the first version we looked at it, what was happening, as Elena just said, for the people who couldn't hear on the microphone, is that the colors are being assigned automatically. And so that way you were getting bins that were occurring in multiple places being assigned different colors. And so this was a manual correction that then just made sure that the bins and the contigs that were assigned to one bin all had the same color. And so you could visualize them all at once, as opposed to being like, I think those are supposed to be what they belong together. And so if we wanted to, we can make this size bigger and we can look at this manually and highlight things and run with that. But we'll move on to the next element of this. So we can X out of that, close out with Command C. Move stuff around. Okay. So now we're going to dive a little bit deeper into the max bin results for this particular data set. And we're going to look at a way to try and start to look at how, if you had this information and the output that you got from the automated output, how would you go about manually refining it to make sure that you had the highest quality bins that you could work with? And so we can do this with Anvi import collection dash lowercase c contigs.db dash p profile.db dash C, big C max bin. So this is like what we did with Binsanity before using contigs mode. We're specifically targeting the max bin results here. So this is going to reset everything, right? So instead of having all of the binning methods displayed here, we're just going to highlight one individual binning method. And then we're actually going to be able to pull out individual bins from within this binning method and look at it at a very fine scale and try and find areas that might be correctly binned or incorrectly binned. And so now we can run this command Anvi summarize to get this information out. It's a lowercase c, sorry. So we're going to output it into something called maxbin-summary. So this is going to run through and it's going to create FASTA files of specific bins. And you can see them being named as it goes. So max bin 0, 7, 007, 001, 003. So these are the 9 bins that Maxbin has determined. And now we've pulled them out as individual entities and we can work with them as individual entities. So when we call Anvi Interactive now, we can specifically call our profile and our contigs. And this time we can work specifically with the Maxbin results. So I'll pause right there for a second. Hit enter. So it's going to redraw it for us. So here we have the bins themselves from Maxbin drawn in that way. So we're looking at, so each of these, the dendrogram now has removed the individual contigs. So if we zoom in, you can see instead of like a paintbrush with lots of little leaves, you just have a single point representing each of the predicted bins. And so you can see up here in the top here. So this is max bin, predicted bin 005. And so we're looking at a really nice, clean representation of the coverage values here. So we're looking at mean coverage. So we can do things like change how we're looking at this. So if we change, so the mean coverage is covering quite a bit of any of the variability. So we can change this to look at, visualize how it's actually being clustered. That didn't change anything. Let's look at abundance, sorry. It's not changing how I thought it would. But what we can do really is look at right here. So max bin 008 has a nice high completion value as estimated by Anvi'o, but based off of this big red block has a lot of redundancy. So if we're measuring how well this bin looks, it looks very complete, but the redundancy here is telling us there's actually a lot of single copy markers that are occurring multiple times. And we can look at this in the bins file. If we generate, nope, that's not what we want. We can't look at this in the bin file. But what we can do is what the next slide is going to show us to do is we can look at these particular entities by themselves and start actually manually pulling out contigs that might be causing this high error of redundancy. And so this is something that is not in the presentation. I don't think we actually get into. So this is a measure really quickly just to cover what this is. So when we look at a genome, we can try and estimate how complete it is based off of how often single copy genes occur in the genome that we're looking at. And so there are a couple ways of doing this. I think the preferred way of a lot of people nowadays is something called Check'em, which is a really great tool to use. It's in the, it's something I will talk about a little bit in one of the other talks today. But what Check'em does is it has a fairly large database of what how many single copy marker genes belong to different groups. And so you can have a very broad, looking at all microbial life, which has a set of 56 single copy genes that almost all organisms have a single copy. And then you can get into very fine detailed situations where you can look at the alpha proteobacteria as a group, for example, and say something like, oh, in the alpha proteobacteria, there are 300 genes that actually are single copy in the alpha proteobacteria that represent that. And so what you can do is you can take your genome bin that you've gotten and ask, how many of these single copy genes do we find? And this gives you an estimate of how complete your genome is. And so that's what's happening here. Anvi'o does it a slightly different way, but in the same similar manner, where it's looking at a set of single copy genes and saying, well, how many of these single copy genes do we have in this bin? And in this case, it says that we have a lot. But then we also know that because there's a single copy, they shouldn't be occurring in multiple copies. And so if we get to the situation where we have single copy genes that are occurring in multiple copies, the assumption is that that multiple copy generally is coming from contamination. So you have contigs from multiple organisms grouped together in a bin that are showing up as multiple single copy genes. And so that's what this red bar is telling us in terms of max bin number 8 here. And we can compare that to max bin number 5, which has a very small, little tiny sliver right here of burgundy, which shows that there's like a little tiny amount of redundancy or error there. And then we can compare this even more so to the next one over, max bin number 2, which has 0, right? There's no color here right there. And then there's varying degrees of this. So this one's a little thicker to show a little bit more redundancy. This max bin over here is somewhere between 8 and 9, or between 8 and the rest of the ones we're seeing. So it's about, maybe half of the bin is contaminated, and we can pull that out. And so we're going to do that. We're going to look at these two slightly higher redundancy bins a little bit more closely. So we can close this out. We go to the next slide. So we're going to use a Python command here to pull out specifically the names that we want to work with for this. So we're going to use a command Python -c. And this is, so if you copy and paste here, I can already tell you that Microsoft or Google or whoever done this has made smart quotes here. So you need to do a single quote, type this out, print, open parentheses, double quote, type out the name maxbin.007, \n to tell us it's a new line, maxbin.008, close quotes, close parentheses, close single quote, and we're going to export that out into a new file called maxbin.bins that we're going to try and refine.txt. So wait a second. I can hit enter. This isn't going to jump too far. So did that work for everybody?
Like it.
Okay, great. And now we can add that information to our profile and specifically just call out those two bins, right? So all we've done really, if we take a look at this real quick, is we've created this file called maxbin bins refine. If we look at it, all it lists is the two elements in the document. That's all we've done there. And that's because what we're going to do is we're going to ask the Anvi'o refine step to find those two names in our database and just pull those out individually. And we'll be able to actually look at, unlike the last clustering tree, which just had each bin as part of it, we'll actually be able to look at the individual contigs and try and look to see which ones maybe are incorrectly binned as part of this. And so we'll type this Anvi'o refine step in. Where will be -p, profile db, -c, contigs.db. This is going to be one of those things where you're like, oh, I get it. That's what he's meaning when he says that. We're going to do big C maxbin -big B, and then the name of the file that we just created, maxbin.bins.refine or -bins.refine.txt. We can hit enter. So now it's clustering just the splits that are part of those two bins. And we're going to generate a new tree based off of that. All right. Okay, so now we have our two bins here. Can you show us that here? No. We can make our taxonomy bigger. So these are just the contigs that make up those two bins. Draw lines. Right, and so these were the contigs that made up those two bins. And you can see right here that despite them being two bins, if we look at even just looking here at the taxonomy side of it, there's actually multiple organisms grouped into these bins right here, right? So we have bin 8 and we have bin 9, but we're gonna have to do some manual work with this to make them into something that we can actually work with. And so we can do that if we click on the bins tab here. Every time we figure out where we wanna call our split, we can actually hit this new bin button and then manually create new bins as we go along. So I'm gonna zoom in. And so you can see there's this one area here that has some abundance along this area. You know, it's high abundance here, high being relative, and then some low abundance in some other samples. It's got a universal taxonomic signature for all of these contigs. And so we can actually really, really zoom in. And we can pick right here, not the one. So if we picked right here, we'd actually capture all this other stuff that's around it, which we don't want to do. We want to pick right here on this part of the split. So you can zoom all the way in. We can highlight right here. So this will highlight all of the elements above it. So we can zoom back out. And you can see that this new bin here is now covering that whole taxonomic range. And then Anvi'o is nice enough to tell us some information about this. So over here, this bin that we've selected is 2.3 megabase pairs. It's estimated to only be 31% complete, right? So for the set of single copy markers that we're looking at, it only has 31% of it. And it has a really low contamination rate compared to what we were starting with, which was, you know, essentially this big giant magenta block that said that we had like almost 100% contamination, which makes sense because we have multiple organisms that we're looking at. So if we're happy with this bin, we can say new bin, right? And now the next selection that we make will have a different color and we can manually go through and assess this. And so one of these other ones that looks really pretty obvious to me is this area here. You can see that the coverage values are pretty consistent across, and it has a really good consistent taxonomic assignment with some issues along the way, but that we can just chalk up to the database and how it was assigned. And so if we zoom all the way in again, we can see there's a major split. Ooh, too far. If we want to exclude some of these ones on the outside here, you can see, I'm going to use the cursor here. You can see they have a slightly different abundance pattern, so we're not interested in them. So if we make sure that we're just starting here, we can zoom in. Okay. We can exclude those couple of contigs here. Come on. Let's get in there. There it is. My computer's just tired. We can zoom back out and look what this bin looks like. So we got most of it. So we might have to go back through and keep adding a couple more pieces to make sure we get here manually. But this bin that we just created is 1.1 megabase pairs in size, and it's predicted to be 72% complete and 3% contamination. So what we've done is we've taken these like really high contamination bins that were created automatically through an automated system, and we're able to visualize it in Anvi'o and use the data that Anvi'o is giving us to actually find the genomes within those bins, right? So while the automated systems can work really well sometimes and give you nice clean bins like we saw on when we displayed all of them, they don't work perfectly. And Anvi'o gives you an opportunity to go in and actually work with them individually in a method to look at that, right? And so we can keep doing this and we can keep doing this until we get all of the bins, right? So this is probably a bin here with a bunch of erroneous extra taxonomic assignments, and we could assign that one. This one's pretty interesting right here, right? So this doesn't have a taxonomy, this stretch right here, but it does have, you know, somewhat of a clear signature in looking at its GC content and its abundance information, right? So this whole range right here might be something. And if we click on it, oh boy, too much zoomed in. So if we click on this area. Let's see. Too shallow. So maybe this area here. Like that. We can click on it. Oh, well, we need to add a new bin. Sorry, first add a new bin. Click on this area. Assign it at that. And so this is a 800 KB, 750 KB fragment that's about 40% complete and about 3% contamination. So despite it not having a taxonomic assignment, here's an organism that we can actually identify that doesn't have a taxonomy and we can work with. And so we can go forward with this and be like, okay, well, why this doesn't have taxonomy? And we can do something like a phylogenetic assessment to try and figure out what this organism is and where it belongs. And so we can keep doing this. We can do this. We can store the information that we generate here as an output where we've now created some new bins. So this does this automatically. Collection max bin is updated in the database. You can close the browser and then we can continue working on this stuff going forward. We're not going to, but this is just to show you how you could do that manually in a manual process. Does anybody have any questions? There's been a lot of blank stares as I go through this at this point. So I'm not sure if it's just because it's like everybody's marveling at how awesome this is and it's really cool and there's all these fun colors that are happening, or if we're looking at it and going, yeah, but why? Why am I doing this? And so if there's a why, that's a question I can try and answer that. Yeah.
Not necessarily a why question, but the trash cans on the side, I'm assuming you delete them, you're not deleting anything in the actual figure itself.
No. So these trash cans, so if you have gone through a lot of effort and you made a bin and you dislike it, the trash can will just delete it for you. And it will make sure you want to do that, and then it goes away. Yeah.
So what makes us think that, like, just in the area where it's not assigning anything, we're like, oh, GC looks relatively equal.
Yeah.
It truly could be something there.
Yeah, I mean, so it's not just the GC that's giving us the hint, it's also the coverage, right? So you can see here, there's this bar of coverage that happens in this one sample that stops pretty abruptly and then hits here. They actually look like they have similar coverage values. And this is probably the reason why MaxBin put them together, right? So they had cover, but you can see very clearly there's a sharp change in GC right there, right? So when you visualize this type of data, this is the type of thing that Anvi'o is designed to do. So that way you can do that 10 times faster and 100 times better than a computer can right now, right? And we're working on algorithms to make sure that you're obsolete in the future. But right now, this is the best way the human eye can really see right away. There's a pattern there that I want to work with. Katie, you had a question?
Why bin manually other than opposed to binning the program?
So the programs work really well up to a point. And even Binsanity, which we have worked tirelessly to make as effective as possible, we still end up with bins that are either high completion but also high contamination, and the computers just can't, the algorithms that we have can't discern why that's the case. And then you can put them into something like Anvi'o, all of a sudden it like falls out. You go, oh, that whole Contig has a GC content that's completely different. Right? Sure, the coverage is the same. And who knows what the reason is, right? Like you could go into lots of biological reasons why that might be the case. Maybe it's a fragment from another organism that, just happened to have the same abundance and that's completely random. Maybe it's a plasmid replicating, a single copy plasmid in there with your organism, right? And, has the same abundance, but it has a completely different GC content. And that might be skewing the results in terms of completion, depending on what genes are on that plasmid. You can really come up with a really long list of things that might be happening there. And so having that little extra bit where, if you're happy with generating a bunch of bins from your data, so a good example with this would be one of the data sets I worked with recently where we generated like 200 high completion, low contamination genomes. And then there was a handful of them, like 3 or 4 that were high completion and high contamination. And then we just kind of ignored them. We didn't do any more analysis on them. But you could use something like Anvi'o to go in and, that data exists in the public. Somebody could go in there and be like, well, let's just try and clean that up a little bit. And, some of the dangers of that is when you clean it up, you split something in that's 100% complete. And you're like, I have a complete genome here. And then you split it up and you get a bunch of little 30s and 40% complete organisms. But that's, that's a risk that you run in that situation. But yeah, so, the automated ones get you really close. They're really good when you have really large data sets with lots of organisms and lots of genomes that you're working with. But then you always need to go over that last little bit of the hurdle where things start to meld together and the computer's not really good at discerning why that's happening. Ooh, 3 questions. I got 3 questions out of you guys on that. That was pretty good. All right, so we can close that up. So if we had saved, I think if we did save it. So if we did open up Anvi Interactive right now with the max bin setting, those new bins should be there. Let's give it a try. It might not work because it's almost lunchtime and the computer's getting tired. I mean, I'm getting tired. So again, another call to Anvi Interactive. Let's cross our fingers and see what it does. Did it highlight new ones? Don't think it worked. Maybe it did. Oh, here you go. So here are the refined new genomes that we selected there, right? So we didn't completely go through and change everything. But here are refined 3, refined 1, and then the one that I was asked so ruthlessly to delete has been deleted. But now we can go back in and we can see where originally there was this big block of magenta that showed that there's a lot of contamination, that we've actually removed that. Let's see. So we haven't fully done that because we didn't make any new bins within genome 9. That's still there. But if we had done that for all of those pieces and redefined what 9 actually looks like, we would have been able to reduce that down and create new bins that work that way. And so that's going to be the end of the hands-on stuff. There's a couple things that we can talk about going forward. So there's just 3 more slides that we're going to discuss. Oh, there's 1 more hands-on. I'm incorrect on this. And so there's a lot, in this particular version of Anvi'o, there was a lot of concern about how do you deal with oversplitting, right? So what happens if you make your parameters so sensitive that instead of calling a genome, you make it into a bunch of different pieces that isn't working properly. So one of the things that we did as part of Binsanity in the automated process of this is we use a biphasic clustering approach, where first we look at coverage and then we look at tetranucleotides and GC content. And that gives you a little bit better preventing that oversplitting from happening. And so that's how we worked around that in our automated system. And so that gives you a little bit more a better way of getting at keeping the bins from being oversplit, right? So taking one genome and making it into multiple bins. In Anvi'o, they suggest what you want to do is change the number of clusters. So in Anvi'o is built in to use Concoct, one of the other automated systems. And with Concoct, you can actually tell Concoct how many bins to look for, right? And so the way you adjust oversplitting with Concoct is you tell it to look for less bins than there are genomes. And so then what you end up having is lumped bins, but because they're lumped and they're actually different organisms, when you go in and visualize them, it's actually easier to just be like, I've lumped these 2 together. It's very clear that they're supposed to be separate. Let me put a split here, and now we have 2 bins and everything's working out fine. So that's how we can, so there's lots of little issues that can come up. The Anvi'o workflow works really well in describing this and how to work with it. The new automated systems are getting better and better. So the need to get into the nitty gritty of individual bins slowly decreases. There's a lot, we don't cover this right now, but there's a lot of questions about you can have lots of low completion bins in your automated output. And if you use Anvi'o, you might be able to stitch things back together, right? We're just spending a lot of time about pulling things apart and making sure we have low contamination. But if you wanted to go for something where you wanted high completion and you had low completion, Anvi'o can do that too. It's a reverse version of what we've been doing here. So in this last slide, so this was an example of that. So we can recluster with Concoct. And instead of using so there's probably somewhere between 8 to 10 complete genomes. If we tell it to identify 5 clusters, this will allow us to visualize it in a way that should allow us to really quickly see how to do that. So we can do that really quickly. It'll be the last thing we do before break. We're going to explicitly tell it that the number of clusters we're looking for is 5. And then we're going to call this a new binning output. called concoct, and we're going to call it -C5 to say that it had 5 elements to it. So that runs really quickly. We can run our Anvi interactive. Hit enter. So what we've got here, if it worked, is, oh, here we go. Yeah. No, that's still there's a taxonomy on it. So what we've done here, we've got a little bit cleaner. I don't think this actually worked properly. I'm looking at Elena. Is that what it is? The bins. There it is. Then concoct C5. Thank you, Elena. So this is gonna, we could compare this to the original if we want to, but we'll look at this first. So here are the 5 clusters that concoct is generated for us. And you can see, right, in this conservative way of doing it to prevent oversplitting, we've actually lumped these 2 genomes together, right? But this again is one of those things where we can really quickly, I got a squinting Sarah over there, so I'll zoom in and prove that to her. So here, concoct with only asking for 5 bins, instead of splitting and creating 2 genomes here, I think there's actually 3 genomes hiding in here, it's just created 1, right? But that has given us a target now that we can go in and manually change. And so if we compare that, I'm gonna just load this really quickly, right? So there are 5 bins here. If I compare that just to the raw concoct output, right? So raw, yeah, that's fine. The raw concoct has 38 bins. And so you can see here, it's effectively split it, but there's a lot more of these little tiny splits in here that are gonna keep contigs from being part of the genome that's actually there, right? So by lumping them together, you can go in and actually do that manual part a little bit as recommended from the Anvi'o people, as opposed to having this really oversplit system that happens just naturally from concoct. But that's it. We are done with talking about binning. Right on time for lunch. Look at that. 5 minutes to spare. Does anybody have any questions before we wrap up and take a break? I know this was a lot. I can tell by everybody's enthusiastic faces that they're like, I am really overwhelmed by that. Please, let's take a break on the next session and just have you talk at me for an hour and a half. And I'll do that. I'm happy to do that as we move on to the annotation part of that. If there aren't any questions, if there's not been any questions from the webinar people, if there were, we could answer that now. But otherwise, we can wrap up. We'll take our stuff with us. It's on you to be finding someplace fun to go for lunch. I'm going to stop live streaming so YouTube doesn't hear.